the inaugural seminar sponsored by the Data Science Initiative at Brown University and entitled Decoding Pandemic Data. We're very pleased to welcome Professor Betsy Ogburn from Johns Hopkins University, and I'll give Betsy a bit more of a formal introduction in a minute. But just a few words. I think we all know that in this COVID pandemic, the knowledge that we're gaining about COVID is being derived almost exclusively by the data that are being generated and posted to dashboards and collected in databases around the world. And these data are telling us about how the virus spreads through epidemic models. They're telling us about prevalence of antibody seroprevalence. They're telling us about drug effectiveness and more. And I expect that we're going to be learning continuously as we go. There are enormous numbers of opportunities for data science in this period of time. And I think that it's really, despite a lot of the tragic side of the pandemic, at the same time, there are enormous numbers of scientists that are committed to not only working in this area of public health, but learning more about things like epidemic modeling, and then bringing skills that are sort of outside the public health sphere, particularly in machine learning, building data dashboards, data visualization. There are a lot of skills that are coming in from outside traditional sources of expertise that I think are going to really lead not only to new knowledge about COVID per se, but to really make the public health data science enterprise better. So this seminar really kind of came about because I was being asked by a lot of data science colleagues, how can I get involved? How can I learn about this? And I have this great idea, who should I talk to try it out? And so it occurred to me that it would be a good time to call on experts around health data science and public health data science to give us a tour of different areas, different topics that are related broadly to how we use data in public health, but now are being applied more specifically to cracking open some of the mysteries of COVID. So the speakers that we've engaged are themselves already heavily engaged in the domain area. And I think this is really a feature of data science that sometimes gets overlooked. We sometimes focus on the dazzling dashboards and the fancy machine learning algorithms, but really the most successful data scientists are those who are not only skilled in the technical side of data science, but also are heavily engaged in a topic area where their skills are being applied. And so I think that this really, one of the goals of this seminar is to really showcase the importance of deep engagement in a domain area to make knowledge about data science have maximum impact. So aside from Betsy, our next two speakers also fit this description. Nick Jewell, who is a professor of public health and biostatistics at Berkeley and the London School of Hygiene and Tropical Medicine. He'll be speaking next Wednesday, and Natalie Dean, who is a biostatistician and an infectious disease expert and a colleague of Betsy Ogburn, will be speaking with us next Friday. You might have, if you've tuned into any of the major networks, you may have seen Natalie discussing all kinds of aspects of COVID, but most recently testing strategies. So the format for today, number one, this session is being recorded, so we will be able to post a video later. We would like to maximize audience participation, so we've asked our speakers to speak for up to 30 minutes. Depending on the audience size and how manageable this is, I think at first we'll start by having audience members send a private chat to me with questions, and then I can just make sure to call on individuals in some sort of order so that everyone will get a chance to ask questions. And if that seems too formal or too stiff, then we can just have folks raise their hand and then I'll call on them. So without further attention to logistical details, I'd like to introduce Professor Ogburn. She's an associate professor, as I mentioned, at Johns Hopkins School of Public Health. Down at Hopkins, as you may know, there is enormous amount of activity happening around COVID and data science. And Betsy is one of our leading experts in the field of biostatistics in causal inference, modeling complex data on networks. And now she and her colleagues are organizing a collaborative space for aggregation of information on randomized trials. And I think today's seminar is particularly timely because the New England Journal just published a large observational study looking at the effect of hydrochloroquine. This is a drug that's been in the news lately, and the best we can do right now is observational studies. And I think even though those sort of studies are near and dear to many of us, we know that they have limited ability to give us the information we need in that in situations like this. So randomized trials, I think, are going to be delivering huge amounts of important information, and they're also going to be supported by the rigor of proper design. And so we're eagerly awaiting a lot of those results. I think some of the work that Betsy's doing is going to help us aggregate this information in effective ways. So Betsy, I'll turn the floor over to you. And I just want to say thanks again for taking time. I think one thing about COVID is that, you know, the folks we're trying to invite to this are already swamped. And that makes it even more generous of you to give your time. And so I really appreciate it.
Thanks, Joe. I'm very pleased to be here. This is a great seminar series. I've been really enjoying the proliferation of online seminars to break up the monotony of working from home. So I'm glad to see this one, and I look forward to tuning into the future ones. Okay. Can you all see my screen now?
Betsy,
gosh,
I think
was I muted that whole time?
Yeah, I'll speak for the crowd and say that your screen is visible here. So I think
okay, but I was muted that whole time. I was thanking you for inviting me and
no, we heard it all. Yeah.
Okay, great. I don't know when I got muted, but great. So yeah, thanks a bunch for inviting me and giving me time to talk about this project. So as Joe said, this project is about the coordination and aggregation of evidence across COVID-19 randomized clinical trials. The project is really a group endeavor. You can check out the website, and on our partners page, we have a lot of people listed who have all played really instrumental roles. I noticed Noam Finkelstein in the audience. He's been doing a ton of programming. If you appreciate the aesthetics of the website, that's all thanks to Noam, who's volunteering his time, and many others. So I really fell into this totally accidentally. I work on causal inference, and for the most part, that means I work on observational studies. Since I sit at a school of public health, I've been involved in randomized trials throughout my time at Hopkins, but it's not at all my area of focus. But sometime at the end of March or the beginning of April, I was asked to advise on a couple randomized clinical trials starting up at Hopkins. And it was immediately clear to me that there was a huge problem, which is that these trials were asking very important questions with likely small effect sizes. They were questions that it seemed any hospital system would want to answer, and they were not in communication with any other teams who might be answering similar questions. And it was clear that they were not going to enroll enough subjects to answer the questions on their own, at least not in a timely fashion. So I set about trying to figure out how we could link up these studies at Hopkins with other similar studies at other institutions, and discovered that there really was no way to do that. And that's how the COVID collaboration platform was born. COVID CP was our acronym before convalescent plasma became the big hope for COVID. So it's a little bit unfortunate that we're duplicating the letters there, but COVID CP is for COVID collaboration platform. So very briefly, and I'll talk more about what the platform is later in the talk, but just to give you the overview before I talk about the background. The COVID collaboration platform is a platform for aggregating evidence across COVID-19 RCTs for similar interventions. The way it operates is basically that it's a repository of RCT protocols that are available for collaboration at various stages and various depths. That could be joining forces with other research teams to create a kind of ground up core protocol that can be used in independent studies of similar interventions. It could be enrolling new subjects from a different site or forming a true multi-site trial from the ground up. And it could be, and we really hope that it will be, sharing data for the purposes of aggregated analyses, preferably aggregated interim analyses, because we would like to, if we're able to aggregate evidence across many independent trials, we would like to be able to get answers sooner than we would be able to if we waited for each trial to completely enroll on its own. So I will explain briefly why a platform like this is needed. I'll talk a little bit more about how the platform works, and then I'll talk about the phases of development that we envision and where we are so far and what we have to do going forward. So first, why is this platform needed? Trials are proliferating across the globe at an unprecedented rate for COVID-19. This is, I update this slide every time I talk about this project, and it's like hundreds of new trials every week. It's just wild how quickly the number of trials is increasing. And this is a fantastic trial tracking dashboard. The website address is here, and you can sort these trials by the interventions that they're interested in, and you can see that there are often dozens or sometimes even more than that trials looking into similar interventions. And most of these trials are not centrally organized or in coordination with other teams interested in similar interventions. This is contrary to the recommendations that came out of a National Academy of Sciences Committee on Clinical Trials following the Ebola outbreak. This committee urged for sharing data, cooperating, and coordinating the global research landscape, but unfortunately that's not happening to the extent that we would want. There have been a lot of commentaries coming out in lead medical and science journals urging basically the same thing, the use of core protocols for randomized trials, coordination across study teams studying the same intervention, limit to single site trials, and other similar recommendations. I'm highlighting this particular commentary. The lead author is Natalie Dean, who Joe said is giving the talk a week from today here. But I'm highlighting this commentary among a bunch of similar commentaries because a lot of the authors are on the advisory board for the COVID collaboration platform, and because this quote really sums up our mission statement. In the context of public health emergencies, study teams should be encouraged to collaborate on existing ongoing protocols rather than starting new independent trials. But in the absence of such coordination, we've seen that local outbreaks can attenuate before a trial that is limited to a single institution or to a single region, geographic region, is able to fully enroll. This happened in China, most famously with a remdesivir trial, but it happened with a bunch of other trials that never were able to fully enroll before the curve was flattened, which obviously is we want the curve to be flattened as quickly as possible, but it's hard to plan for trials when you don't know how long you'll be able to enroll patients for or exactly what the volume of patients will be. Lots of small and single arm trials will never be able to provide reliable data on their own, but they could meaningfully contribute to a body of data. And then there's a real risk not only of false negatives with underpowered small studies, but also false positives. So underpowered studies have a higher probability of type 1 errors or false positives, which is not always recognized in the literature. And then I would also argue that we have essentially a multiple testing problem when multiple trials are examining similar interventions at the same time for the same global research environment. That it's not a single researcher testing multiple hypotheses at the same time, but it's essentially a multiple testing problem just divided up across multiple research groups. And this gives an increased risk of at least one type 1 error across the collection of studies. And you can imagine if you combine this multiple testing problem with the file drawer issue, that we might see, if it turns out that hydroxychloroquine has no effect, we might, and there are 100 trials testing for an effective hydroxychloroquine, that we might see 5 false positives that make it into the news headlines and totally change the way treatment decisions are made. So we are very much not the only ones working on these problems. We're one group among many trying to coordinate disparate data sources and aggregate evidence for COVID-19. But what sets the COVID collaboration platform apart from other endeavors is, first of all, our focus on randomized clinical trials. And second of all, our focus on building collaborations along the way rather than aggregating data after the fact. Lots of other platforms are efforts to combine and publicize COVID-19 data, but they're generally focused on collecting existing data and not building active collaborations. And they're mostly focused on EMR and other observational data sources. And then there are lots of large multi-site trial coordination efforts, both in the US and around the world. So Solidarity, REMAP-CAP, Active is the NIH pharma partnership. But these collectively, these cover a subset of all of the interventions of interest that are being studied out there. And they also don't encompass all research teams studying those interventions. And then there are lots of registries like the trial tracking website that I pictured a couple slides above. Those keep track of trials and are really useful for exploring the landscape, but they don't actively support coordination or collaboration. So just to emphasize, we really want to fill a void. We're not at all interested in duplicating efforts. And if somebody else came along and wanted to aggregate RCT efforts across COVID-19, I think we would be very happy to cede to an existing group. We exist because we saw a void, and we'd be very happy to have that void filled anyway. Okay, so how do we fit in? The purpose of COVID-CP is to bring disparate research teams working on the same question, mostly in the context of RCTs, together to share protocols, data, and other resources. Ideally, we would like to catch studies as early as possible so that they can work together on a shared protocol. So we would ideally like to catch research teams when they're just starting to figure out their trial design and they have a research question in mind, but not necessarily a full protocol, because the earlier we can catch studies, the more syncing up we can do and the easier it will be to aggregate evidence. So the ideal would be that study teams can work together on a shared protocol and possibly form an actual multi-site trial under a single PI or set of PIs. But if the logistics of a multi-site trial prove impossible, then we at least hope that study teams will approximate one as closely as possible. So for example, running parallel trials with protocols that are as similar as possible and sharing as much data as possible along the way. And we hope that this will solve several of the issues that we pointed to. So if protocols are public and available for collaboration, then data can be collected by different teams in different geographic areas as the epidemic travels. And aggregating evidence across multiple sites will get us answers about what treatment and prevention strategies work much faster and more reliably than analyzing subsets of the data in isolation. And it will also avoid the type 1 errors error issues that I mentioned. Overall, our fundamental goal is to get reliable evidence into the hands of policymakers and clinicians and the general public as quickly as possible. The primary goal here is not the usual academic products, although that can obviously be a secondary goal. Okay, so how exactly does this platform work? PIs can submit a draft or a completed protocol. And here's a screenshot of the subpage on our website where you would do that. And then they can receive requests to collaborate on that protocol, and they can accept those requests. Researchers can also click on the request to collaborate button for a particular protocol and request to collaborate. And as I said, a collaboration could mean any number of things. So we want to be really flexible about what collaboration means so that we can both encourage maximal collaboration, but also support more limited collaborations, recognizing that asking people to be completely open with their protocols and data or to form multi-site trials on the fly might be impractical. We want to be able to support whatever level of collaboration researchers are up for. So collaboration could mean working together on a draft protocol to create a mutually agreeable core protocol that could be unrolled at multiple sites. It could be a request to join a study as a new site or to enroll subjects under the original PI. And then it can also mean, and we really hope that it will mean, sharing data for aggregated analyses to pool evidence across sites with a similar research question. Again, preferably interim on an interim basis, but at least at the end of the study. When we receive a protocol submission or a request to collaborate, we vet it very minimally just to make sure that essentially that it's not spam. We make sure that it has legitimate scientific value, that a protocol has a well-defined question and well-defined interventions and outcome measures, and that a request for collaboration is coming from somebody who really is in a position to perform research and is not spam. Anyone using a posted protocol has to agree to cite the original PIs so that they receive credit. And then we will keep track of collaboration requests and any active collaborations and then support them as much as we can. So we're lucky that the Trial Innovation Network, the SMART IRB, and Vivly.org have all agreed to provide support. Vivly is providing free data anonymization and data storage for any data that researchers are willing to share. And then we're providing free statistical support, mostly because statisticians have been volunteering their time and will continue to volunteer their time to either perform or advise on pooled analyses. So here's a screenshot of the How It Works page on our website, which basically summarizes what I've just said. And now I'll talk a little bit about what we've accomplished so far and what we are still working out. So phase 1 is essentially complete. We've launched a platform where researchers can submit new protocols and can search posted protocols to find existing studies that match their research question. We have support for collaborations and for aggregated analyses available through Vivly and through statisticians who are volunteering their time. We currently have 12, actually make that 13 studies that have posted their protocols. We have a few more that we expect to come in. We've received 1 request for collaboration. 3 of the studies that are posted are actively looking to find partnering sites to enroll more subjects because they're currently single site trials and they know that they will not be able to fully enroll that way. And it looks like we will be able to do matchmaking for that and provide them with collaborators to enroll more subjects, although that's not totally finalized yet. And then we're just actively trying to get the word out to researchers that this platform exists, trying to build trust in it and encourage participation. And then the next phase, which we're just now beginning, is to move towards data sharing and data aggregation. So we're working on recommendations for common data standards and metadata. We're developing analysis plans for interim and post hoc analyses and putting together some recommendations about minimal data elements that we can strongly encourage all participating researchers to use in their studies. And then, as I said, so Vivly is, Vivly.org is providing free data anonymization and data storage. We're looking into other possible options for data deposition platforms or alternatives like containerized analyses. I guess we want to figure out the best balance between allowing researchers to maintain control over their data and also allowing whoever performs pooled analyses to have access to the most informative set of data possible. So we have one system set up now, and we're trying to figure out whether that's the best long-term solution or whether we should consider other alternatives like distributed analyses. And then in the third phase, where we actually perform aggregated analyses, especially interim aggregated analyses, we're very concerned about the right level of oversight for that. So we're talking right now with ethicists and trialists and clinicians about the idea of having a kind of overarching data safety monitoring board that would oversee these interim analyses and communicate with the trial specific DSMBs. It's kind of thorny, you know, pooling data, sharing data and pooling data from independent studies before those studies have completed is against the existing norms. And so we really want to make sure that if we're going to recommend doing this and facilitate doing this, that we do it in a way that is ethical, is doing right by patients who are sacrificing their resources to participate in a trial, and is maximally informative, because the whole point is to get good information out to decision makers sooner. So we don't want to sacrifice informativeness any more than we have to. We're putting together a group of data scientists who will actually be able to perform these analyses. Hopefully we're getting funding so that they don't have to continue to volunteer their time indefinitely, but we'll see about that. And then we plan to continue to try to develop the needed infrastructure and support for collaboration as we discover new ways to improve along the way. We want to be able to move really quickly and nimbly to adapt to the needs of the national and global research community. And finally, we really hope that something like this collaboration platform, hopefully more functional and with greater buy-in, will be in place in time for the next global public health emergency, so that this type of coordination can happen from the beginning. And then this is the last slide. I just want to close with some of the things that we've been struggling with in trying to promote this new mode of collaboration. So academic incentives and norms are really aligned against this kind of on-the-fly collaboration. Multi-site trials usually take months and months, up to a year, to set up. And data sharing is very much outside of the norm. And we've heard stories of data monitoring committees that are really against it. But I would argue that that in this kind of setting, research ethics really point towards this kind of collaboration and away from the typical MO, research MO, that it shouldn't, I would argue that it shouldn't be permissible to run a COVID-19 trial without taking the full research landscape into account. For example, it shouldn't be permissible to run 10 similar trials in parallel without coordinating them, because this is a waste of resources. It fails a duty to patients to make the most of their participation in a trial and to muster all possible evidence in assessing clinical equipoise, and not just the evidence coming from your own isolated trial. And then it's also, I would argue that it's a multiple testing problem, that in this setting where like the whole global research community is focused on a similar set of questions at the same time, it's really, yeah, I would argue that it's unethical not to consider all the other studies going on at the same time when deciding to run a single site study or deciding a stopping rule, for example. But on the other hand, there are major new ethical considerations that come into play when we build these collaborations on the fly. So most importantly, how does oversight over aggregated analyses interplay with site-specific oversight? And so we're actively working on these questions. And I should have said at the top of the talk, but I'll just say it now that we are very, very much open to all feedback and criticism and input. We would love for anybody with an interest to get involved. If you know PIs running COVID-19 trials, please encourage them to participate or to give us feedback. Yeah, we're very open to feedback from all comers. And that's it. Thank you.
Okay, Betsy, thanks so much. I think we, it's actually, you know, I thought before you started describing this, that this seemed really complicated. And now I think it really is. There's really a lot to it. So we're gonna open the floor for discussion and questions. So I don't, let's see, I originally said that folks should send me questions by private chat, but I think that the audience size is manageable enough that we can use the raise hand function. I'm looking at my own panel though, and I don't see it available. And I wonder, yeah, now I'm hearing from some folks out there that the raise hand function is not operational. So I guess I'm going to turn to Alden just for a moment and ask Alden, is there a way to enable that?
I am looking.
Okay, so we'll just, looks like we have, let's see.
Nope, that's thumbs up, thumbs down, not raise hand.
Okay, so we do have, I'm seeing them now, so all right. So some folks have discovered it and others who haven't yet. I think we got some instruction from one of our participants. So I'll go to, let's see, I just saw a raised hand that now just went down. Okay.
That was me. I'm just the guy that told you where to find the raise hand function.
I got it. Okay. Okay. So I'll open the floor to see if anyone wants to ask a question. I have a few here, but I want to open to the audience first. Okay. Well, why don't I lead off here? So Betsy, you know, I think a lot of what you described was the the analytic structure. I mean, not the analytic, the sort of infrastructure of getting these together. You know, what sort of like, can you say briefly like what sort of analytic method you're contemplating? Because you know, just take hydrochloroquine for example. I guess you're probably going to be dealing with a handful of trials on hydrochloroquine if you know, those running these trials want to send their data to you. I mean, I don't have to explain to you, but just some of the ideas that come to mind, or some of the issues that come to mind are, you know, you have different populations. These might come from different countries. One might reach for things that we find in meta-analysis as a first pass, but are you going to have like sort of a global protocol for analysis? Or are you going to build off the individual trial protocols? Like how do you envision that happening?
Yeah, that's such a good question. And I'm not sure that I have good answers, but I'll tell you where we're at right now. We just have no idea whether we'll ever get data and what kind of data we'll get. So we're being and also how much, whether sites will want us to perform the analysis or to give them advice or just facilitate pooling data that they can then come together to analyze themselves. We just have no idea what this will look like. I think our ideal would be that we catch trials early on, we get them to collect a similar, we get them to define interventions similarly, similar is obviously vague, but as similarly as possible, and then collect some really objective outcomes. One of the challenges is that there are these outcome scales that have been proposed. There's one that's a 10-point severity scale. There's a 7-point severity scale. It's unclear what the best recommendation there is. Our way around that so far has been to recommend collecting a set of, I think it's 6 binary, fairly objective outcomes. So presumed diagnosis, hospitalization, ICU admission, supplemental oxygen use, ventilator use, and death. And those are components of these 5-point scales, or some of them are, we can back out from the 5-point scales, but we're currently asking that every trial that participates has at least one of those 6 outcomes in its roster. And so we feel like having an objective binary outcome will, even if it's not everybody's favorite outcome, will at least facilitate these analyses. And if our sample size is big enough, I think what everybody cares about is mortality, but often sample sizes are not large enough to actually use that endpoint. And that's where these severity scales come in. They're more sensitive to smaller effects. But hopefully, in an ideal world, we would have large enough sample sizes that we could just go directly to mortality in every study. And yeah, we're considering prospective meta-analysis. We're hoping the ideal case, again, would be that we get detailed patient level data from every site. But we just don't know. So
Okay.
yeah, I think the answer is we're trying to prepare, and we're at the early phases of preparing for all possible scenarios while
Right.
hoping for the best one.
Right. Okay. We hear that
I
a lot
also-
lately. It's like,
Yeah, this,
plan for the best, prepare for the worst kind of thing.
right.
Yeah.
This may not be fair, but we've also just become so mired in the logistics and sociological aspects of trying to encourage people to participate that it feels like, especially because most of the people involved in this are statisticians, it feels like coming up with a statistical plan will be really easy once we figure out everything else. I'm sure that won't be the case, but it's been easy to put that off and spend more time working on the other details.
Right. Well, as someone who has reviewed, clinical trial proposals, the statistical analysis plan is usually like 2 pages, and the protocol itself is like 700. So it tells you where a lot of the effort goes. Okay, so I see that John has a question. Why don't we go to John?
Am I supposed to just speak or?
Yes. Oh, it's Jan. It's Jan Steingrimson.
Yeah, sorry, I didn't know Zoom was going to be popular, so I did my username too early. But yeah, thanks again for a great talk. Really, really interesting for sure. I have 2 questions, both pretty short. The first one is, can you explain a little bit the benefit of adding a DSMB beyond just what the NIH is doing, or the NCI, or wherever this is coming from? Like, why add an additional layer on top of already existing sort of infrastructure?
Sites. Well, so there is no infrastructure from on high. Every site has its own DSMB. Is that what you mean? Or are you talking about like the NIH active?
Yeah.
Okay. Yeah. So, right. Active was announced, I don't know, maybe 2 or 3 weeks ago now. And it's a partnership between NIH and I think 13 pharma companies to study. It's unclear what they're going to study, but it seems like they're probably going to study like a handful of novel drugs. And the reason that doesn't obviate this platform is, first of all, it's not clear that academia is really involved in that. Academics are involved in some like committees to try to decide how this partnership works, but it doesn't seem to include academic research. And then it seems like there isn't a ton of information available, so this could all change, but it seems like they're only going to focus on between 6 and 8 investigational drugs. So that will not cover the world of like 100 different interventions that people are interested in studying. And then the third reason is that it's not really off the ground yet, and we don't know when it's going to start.
Sure. Yeah, that makes sense. Thanks. On the role of the interim analysis, do you see it mostly as a futility thing, or do you see it as an efficacy, or?
I think it should be both, right? Like ideally, if there are 100 trials studying hydroxychloroquine right now, as soon as the collective sample size is large enough, we should figure out what it says and stop running those trials.
Do you envision, like, because people get funding to do 100 people trials, and once you start cutting the funding earlier, do you think that's a barrier to
Definitely.
people?
Yeah, we're really trying to figure that out. At first, our thought was that we would try to get researchers to buy into these interim analyses, but we would have some kind of, and we would make the results public for decision-making purposes, but we would have an embargo on like publishing anything using them. The thought being that you could continue to run your own study as you liked. But then we realized that that's really unethical. Like if we find a safety issue or make a decision about efficacy, you can't keep running your own trial. So yeah, that's where we're trying to figure out. So the idea would be that we run an interim analysis, report the results to our overarching DMC or DSMB, and then they would communicate, the DMC would communicate back to the individual PIs and their own DSMBs. And then I think the idea would be, but please weigh in on a better way to do this, that the site-specific DSMBs would be able to make whatever decision they want, but they would have access to, you know, presumably if the overarching analysis showed a strong signal, the site-specific DSMBs are going to take that into account and recommend something based on that overarching analysis, but that we wouldn't dictate what type of decision they have to make. But you're right, it probably would end up in cutting off studies before they finish. And I don't know, it seems like that's just the right thing to do in this setting, but it really works against all current incentives.
I agree. It sort of points out an inefficiency in the whole system.
Yeah.
I agree with you. Thanks a lot.
Thank you, Jan. One thing I'm kind of noticing in COVID is that we're discovering a lot of inefficiencies along these lines. You know, when when decisions have to be made yesterday, you do start wondering, you know, why should I have 2, 3, 4 small trials trickling along when I could combine the data and maybe get some more sort of precise answer? But it's a tricky issue because investigators come at things from their own angle and they think that they have the right way to do it, and maybe their way is sort of better suited to a particular setting. So these are as you, I think you used the word thorny. These are definitely thorny issues. We do have a question on the chat from Huanhee Hong, who asks, can a trial that will be created via this platform be registered to the NIH?
Oh yeah, for sure. This is not meant to obviate any other registry or data sharing platform. There's nothing exclusive about this at all. In fact, one thing that we're doing right now is going through, we have a team of med student volunteers who are going through clinicaltrials.gov and other trial registries and finding, like kind of surveying the landscape of registered trials to pick out protocols that we think are high quality and that we would like to put up on the site to encourage participation in them, and also just to figure out what's the landscape of questions that are being asked, and can we find questions that could benefit more from this kind of data aggregation than some others, and try to target those PIs. So we're very much using clinicaltrials.gov, and definitely not, there's nothing exclusive about participating in COVID CP that would prohibit you from participating in any other endeavor.
So would a trial that is analyzed or sort of included in a COVID CP project, would that be registered as a separate trial, or would each trial be registered and then you would do the aggregated analysis? Like, how should we think about this?
Yeah, it depends. So right now, it seems like the few collaborations that we think we will be able to really, that we see now as becoming pretty confidently becoming fruitful collaborations, are going to be independent trials that have similar protocols and similar data elements, and we do aggregation centrally across independent trials. So those would then be registered separately. In fact, some of them already are registered separately on clinicaltrials.gov.
I see.
But we also we also have some trials that would like to become multi-site trials, but have not been able to find other sites. And so for those, they are registered on clinicaltrials.gov, and their goal is to find other sites to join their specific trial. So then those sites would not register separately. They would become a new site under the existing trial.
Okay. So if you were to submit something, say, to a journal, again, for those who are not familiar, you know, the thing about randomized trials, for those who aren't familiar, it's not necessarily as simple as just tossing a coin for everybody that comes in the hospital. I think that most of us appreciate that. But what many of us who are not familiar with trials may not appreciate is the many, many layers of ethical approval, institutional review board approval, and the need to carefully document really every single logistical step in a trial, from how the drug is manufactured to how it's administered to who administers it and where, how to define the endpoint, how to record the endpoint, and so forth. Not to mention how to analyze the data once it's finally collected. So, I mean, just to like briefly, if you were to submit an analysis from several of these trials, would you think of like a blanket protocol that covers all of them and attach all the other protocols? Like, is it? I guess what I'm saying is it a trial that you'd be submitting, or is it an aggregated analysis that you'd be submitting? Because I think those are probably different things.
Right, and I think it depends. That we would like to be able to facilitate both of those models.
Okay.
I thought you were going to ask a different question about like giving credit to all the different teams in publications, which is another thorny issue. That
I do want to get to that. I have that question actually written here, but somebody, Sona Akopian, submitted a question on the chat. I just want to make sure that you get to hear this question. It says, would the data collected from these trials be available to the public?
That's a really good question. We certainly think that it should be, at least at some point, but we're trying to be as flexible as possible and allowing individual researchers to make those decisions for themselves, because we don't want a requirement to make your data public to scare people off from participating in an aggregated analysis. I guess it's public data would certainly be the best case scenario, but we still feel like if somebody's willing to share their data with a centralized team that's really thought very carefully about the best and most ethical way of running these analyses, that's still better than not sharing at all. So right now, this is one of many areas where we have a recommendation, but not a requirement. So right now, the way data sharing works is through Vivly, and Vivly's data is always behind a slight wall. So you have to prove that you are, that you have a genuine research interest in the data, which I think means being affiliated with a research institution of some kind, and prove that you have at least a minimal statistical plan for what you're going to do with the data. But I think individual researchers can tighten that wall if they want to, and we're not going to prohibit them. But we are in favor of as much transparency and public access as possible across the board.
I think this is, you know, people who work in these trials probably understand this tension. And it's, you know, on the one hand, the sheer effort that goes into collecting these data, I think my own experience or my own sort of view of this is that those who go through all this effort to collect the data feel a certain ownership over those data, or a certain, maybe ownership is the wrong word, maybe the right word is stewardship. And so I think that it probably makes sense for those who designed these studies and collect the data have some sort of influence over how the data will be used. And as you say, it sounds like in a lot of trials there can be like, especially federally funded or publicly funded trials, there can be a mechanism by which outsiders, when I say outsiders, outside to the process, outside researchers can can make requests of the data, but they'd have to spell out exactly what it is they want to do, and then maybe agree to collaborate with those who collected the data and so forth.
Yeah.
So it's funny. I think that there was a time where I felt personally like everything should be transparent, just throw all data sets on the internet. But then, you know, after seeing a lot of this stuff up close and how much blood, sweat, and tears go into collecting these data, and also the potential for misuse for complicated data sets, it does make sense to have some sort of control. But this is definitely, especially in the era of COVID, it really brings a lot of these issues right to the surface. You know, the issues of multiple trials or data sharing, a lot of these things that we've kicked around for a while, I think in public health, are now taking on.
Yeah, they're urgent now all of a sudden. Yeah.
Well, this might be a segue to the question that you sort of guessed that was going to get asked is, so the broader question was, what's the incentive for participating? And I think there's an altruistic incentive. I think there's an incentive that comes from pure, the desire to get the scientific information out there and have it done in a proper way. But then, you know, again, on clinical trials, again, I'm sure you know, just getting authors in the right order can be a week-long process. How is it that authors or PIs from 5 or 6 trials now are going, or have you anticipated this issue, and what magic are you going to work in order to apportion credit?
Yeah, we've thought about this in a short-term way and a long-term way. Short-term, I think we have to rely on altruism and hope that people are willing to make a sacrifice on the academic incentive side in order to save lives in this emergency. But then long-term, I've gotten a lot of feedback when I talk to clinical researchers about the incentives issue, lots of feedback about what sorts of changes would need to happen long-term. The biggest one is getting data sharing credit and having that become a norm. So a couple people have suggested that that a line on the NIH BioSketch should be, how have you shared your data in the past, and like what research products have come from your data without you being involved in them, to show the value. You know, the idea being that NIH, and I guess this would go for other funding agencies as well, but that funders should want to fund the collection of data that's going to be maximally useful. And so a measure of a PI's previous success is how useful was their data beyond just what they had proposed to do with it. Then there have been suggestions for like adding data authorship credits to be tracked through your ORCID ID. So when a research group uses data that they didn't themselves collect, there's some way of attributing that, a standardized way of attributing that in publications that then gets tracked the same way citations do. So it's somewhere in between authorship and citation. It's more than getting a citation, but less than being an author. So data authorship is the term that at least one proposal, this was a proposal that was published in like NEJM, I think, several years ago, but is especially relevant again now.
These are very innovative ways of thinking about it. You know, because it's an issue that will have to be, I think, cracked open. But I guess I hope that maybe this also, for those who are attending or wind up watching this, especially maybe those who are outside the public health sphere but have very strong interest in data science and wonder why aren't data just posted. Collecting data is really hard work, and there's really no two ways around that. So, but I personally I applaud your group trying to find ways to break some of those log jams, because I think there are data behind very strong firewalls that could be valuable. At the same time, we don't want to convince people, or we don't want to force people to say, well, if you work with us, you have to post your line-by-line data on the internet. I mean, I think that's a massive disincentive. So finding a middle ground could be hugely valuable. I think that's terrific. Okay, one of my personal complaints about Zoom calls is that they go right to the top of the hour and then there's another Zoom call. So I don't want to repeat that mistake for the folks who have attended. And I want to be able to show appreciation for them being here. Betsy, I'll just ask you to stay on for a couple minutes if you don't mind.
Sure.
And for everyone else, thank you so much for attending, and we'll be announcing the next one soon. Thank you again.
Thanks, everyone.